acids to create the proteins. Essentially, how this happens is the code from the DNA, which is basically the house of all the genetic information for your cell, is transcribed into the RNA, which takes it out of the nucleus and into the cytoplasm, where, with the help of ribosomes, it creates proteins. This entire process is called the central dogma. It occurs, like I said, in 2 stages. 1, the DNA is transcribed into the RNA, and this is called transcription. And 2, the RNA is used to make proteins. This is called translation. Let's take a look into transcription. Essentially, what we have here is DNA. Okay? And in this DNA, you have a little section called the gene. And this gene has a starting point and a stopping point. In the starting point, you have a thing called a promoter. Whoops. And basically, what this promoter does is it attracts an enzyme called RNA polymerase. So this enzyme, RNA polymerase, is very essential to transcription. So, what happens is you have the gene, right? And then RNA polymerase attaches to the promoter. And this is the enzyme. So what this enzyme does is basically it splits the two strands of DNA for a moment, and they rejoin at the end of the enzyme. So you have 2 types of strands here. One is going to be called the template strand. And it is called this because this is what the RNA is going to use to create a copy of the DNA. So this upper strand is essentially where the main code lies. And this template strand is basically the opposite of that code. What RNA polymerase does is it creates another strand of messenger RNA to copy the code or the opposite of the code on the template strand. So the opposite of the code of the template strand is essentially going to be the same as the code in the non-template strand. So basically what this mRNA does is it copies the chain in the gene and it leaves. So now you have, by the end of transcription, a regular code of DNA plus a strand of mRNA, which now has the code for creating the protein. This strand of mRNA has a 5' cap and a 3' poly A tail. Essentially what these two do is protect the RNA from other enzymes in the cytoplasm. So this strand of RNA leaves the nucleus by diffusing out of it through nuclear pores. and it enters the cytoplasm where ribosomes attach on it, and it begins the process of translation.